In this video, we'll look at the determination of root epidermal cell fate. There are two possible fates for root epidermal cells. There's hair identity and non-hair identity. Now, it isn't immediately evident when fate has been determined if all you have to look at is a picture of the root. So for instance, here is the proliferation zone, the root tip. This is where the cells are dividing quickly. Above it, so later on in the development of these cells, is the elongation zone, where the cells become more sizable. Beyond that, and so a set of older cells yet, you find the maturation zone. And in the maturation zone, that's when the actual hairs start to form from the cells with the hair cell identity, with the hair cell fate. So you have your root tip, which is the proliferation zone, the elongation zone behind that, and only in the maturation zone can you tell what the fate of these cells is. And yet, as I'm about to show you, the cell fate is determined, decided, as early as within the proliferation zone at the root tip. Now, in order to study fate determination, scientists looked for mutations that rendered the plant incapable of a normal pattern of epidermal cell differentiation. What is that pattern? Well, you'll notice that not every cell is a root hair cell, not every cell is a non-hair cell. You'll also notice that the columns of cells that are colorized here show us that all of the cells in a particular column, and those are all in the same lineage, meaning they all have arisen from the division of the same mother cells, these cells all take on hair fate. Each one of those cells makes a hair. And in this column next to it, these red colorized cells, none of those cells become a hair. This is the pattern. Columns of cells, either hair identity or non-hair identity. When you want to study a process, you very often look for mutations in which the process doesn't work correctly. Typically, if you're looking for something, how something works, you look for when it doesn't work. And what was expected in this gene search was that they would find roots that lacked hairs altogether. Surprisingly, the mutation that was found, the first one that was found, and the only one that was found for quite some time, was the GLABRA2 gene, which, instead of lacking root hair cells, lacks non-hair cells. Every column, every cell on the epidermis of a GLABRA2 loss-of-function mutant forms a hair. They were called hairy root plants originally when found, and they are the result of GLABRA2 loss-of-function mutation. Now, subsequently, people found plants that lacked hairs as well. So all of these have the non-hair identity. And there's nothing else different about these plants. So these are genes that are very specific to the formation of hairs. Well, I say that nothing else is different. Nothing that isn't hair-related is different about these plants. So why would some cells on the epidermis form hairs and others not? Well, differential gene expression. But what causes that differential gene expression? Let's look at the pattern of gene expression of this GLABRA gene. Remember, when GLABRA is missing, you have every epidermal cell form a hair. Therefore, GLABRA's job must be to stop hair formation. Therefore, if you think about it, what this means is that the default condition for epidermal cells, if they don't get some genetic signal saying otherwise, their default condition is make a hair. When GLABRA is missing, they make hair. So GLABRA is the signal don't make a hair. In this picture to the right, you see a set of cells that represent cells that express glabra, and yet glabra doesn't turn cells blue. These cells arise in a plant, otherwise wild-type plant, in which a glabra reporter construct has been engineered into its genome. So you use a glabra regulatory region, you use a GUS coding region. GUS is the protein that turns cells blue. Only those cells that express glabra will express GUS, and so that is our evidence that these are the cells that express glabra. So in a wild-type plant, these are the cells that will not form root hairs. The cells that don't have glabra, right? That's the default condition. Cells that don't have glabra do form root hairs. So each one of these white epidermal cells will form a root hair. Each one of these blue cells will not. Point out that this outer layer is the root cap, present only at the very tip of the plant, and this is a picture of the very tip of the plant. This is a picture of when the determination of cell fate occurs, because that's when glabra is expressed. And again, only much later in this cell's lifetime does it actually begin to form a hair or not form a hair. So, the blue cells are those that don't form a hair, the white cells are those that do. The blue cells, it turns out, are those that are adjacent to only one cortex cell. Notice that if there are two cortex cells in contact with one epidermal cell, that cell becomes a hair. If there's just one cortex cell adjacent to a given epidermal cell, glabra gets expressed in that cell, and it doesn't form a hair. We now know two of the genes involved in the expression or lack of expression of the glabra gene. So glabra is a transcription factor. These, CPC, which stands for caprice, and WER, which stands for werewolf, are also transcription factors. Remember, transcription factors are typically proteins, and proteins have to be expressed. So when the glabra gene is turned on, it can only be turned on when werewolf is present in greater quantity than is CPC, because werewolf and CPC, werewolf and caprice, compete for sites on the regulatory region of the glabra gene. When there's more werewolf, werewolf is the one that binds to the gene. That leads to the expression of the gene through attracting this RNA polymerase to the gene, leading to the transcription and translation of the gene, leading to the glabra2 protein being present in the cell. And notice what's happening. The glabra2 protein is then going off and shutting off the gene expression required for formation of root hairs, right? All of these genes would be on if it wasn't for glabra presence. So it's shutting off root hair formation. In the other column of cells in a wild-type plant, they express more caprice than they do werewolf. Caprice outcompetes werewolf. Now you get a different set of proteins on that regulatory region, one that is not friendly to the RNA polymerase, one that doesn't lead to RNA polymerase binding, and so no glabra gets made. And if no glabra gets made, hurrah, hurrah, all of the various genes involved in making root hairs are allowed to be expressed, and roots are made. How do we know about CPC in werewolf? Well, scientists discovered more loss of function mutants later on. Now, werewolf looks just like glabra in terms of its root hair phenotype. So when it's missing, it looks as though glabra's missing. So when it's missing, glabra isn't expressed, and you get a hair on every cell. When CPC is missing, well, that means werewolf outcompetes CPC in every cell, not just those in a subset of the columns of cells. So CPC is missing, werewolf always gets to bind, glabra's always turned on. When glabra's always turned on, you don't ever get hair formation. I told you earlier that language, the language of transcription factors was important. And so here, in the context of a root epidermal cell, there's a bunch of transcription factors that allow the kinds of proteins that are made by root epidermal cells to be generated. Now, here's my evidence that a transcription factor can mean something different in a different context. Here is a picture of a leaf. This is a leaf with that same glabra2 Gus construct. You'll notice that the hairs, they're called trichomes in these leaves, these hairs all have Gus expression. When Gus is missing, you get, when glabra is missing, you get leaves lacking hairs. Exactly the opposite phenotype. Once again, glabra is involved in the formation, or whether or not hairs form, but in this case, it does so in the opposite way. A glabra mutant has very hairy roots and hairless leaves. Glabra means hairless. That's how the gene was named.